Hey everyone, this is Gayatri and I'll be giving an overview on our pilot project of wastewater detection of SARS-CoV-2 outbreak in the campus dormitories. This project was initially started by the Michigan Department of Health and Human Services along with the Michigan Department of Environment, Great Lakes, and Energy. Their main objective was to confirm the presence of the virus and identify the trends to stop the spread of the virus. The overall goal of a project is to detect the virus which is shedded in human waste of an infected person. The study has been done on 7 major GVSU dormitories. Moving forward, here I will be talking about the daily process that we carry out in the lab once the wastewater comes in. We receive the wastewater from the manholes that are located near the GVSU dorms. Once the wastewater comes in, we add sodium chloride or NaCl and a chemical called polyethylene glycol or PEG in it. After that step, we let it rest for about 2 hours on a stirring machine. The stirring ensures the complete dissolving of the salt and the polyethylene glycol. After the 2 hours are done, in step 2, as it is mentioned, we give it a nice long spin of 45 minutes and the process is called as centrifugation. This step is done in a machine called the centrifuge machine. The third step is a very critical step, that is the viral RNA extraction. RNA is a type of nucleic acid, also called as ribonucleic acid. The RNA is the building block of the virus, meaning the virus is made up of RNA. The fourth step is the detection using ddPCR. I will be talking about ddPCR in detail in my further slides. All the mentioned steps are done by wearing a personal protective equipment, a lab coat, a mask, a pair of gloves, and a face shield. The bottom right is the ddPCR machine. ddPCR is digital droplet polymerase chain reaction. Polymerase chain reaction is like a copy machine of biologists. In the image of ddPCR, we see that there is an automated droplet generator, and when the oil which is already present in the machine and the sample come in contact, there are spherical particles created or droplets. Then the samples are shifted to the thermal cycler machine or the copy machine, and then the samples or the droplets are analyzed for the presence of the viral RNA. Once the thermal cycler is done, we shift the samples to the digital droplet reader. Here, by reading the online signals, the droplet reader will identify if there is any viral particle present in the sample. These are the compiled results after the ddPCR. Here, the letters G1 to G7 represent the dorms that we are working for. Each colored bar here represents the date from which the samples were processed and the results were obtained. We see that 1000 is here like a baseline, which indicates that results at 1000 are the negative results, but anything above 1000 are the positive results, and there is a presence of the N1 gene copy that is above 1000. As this is a pilot study project, here are a few of the mentioned challenges that we faced at the beginning of our internship. Challenges regarding to the optimization of the protocol and determining the general workflow for the laboratory. Further, how the data can be analyzed and predicted using ddPCR was also another major challenge. Lastly, I would like to thank the mentioned staff and faculty for their constant support and guidance and who has kept us going with our good work till now. Thank you.